I am very grateful to use this opportunity given by Kalpagam to introduce Dr. Hari Krishnan sir. Dr. Hari Krishnan VS, BVSc and AH, MLAS, working as scientist in charge in Division of Laboratory Animal Science, Biomedical Technology Wing, Sri Chitra Tirunal Institute of Medical Science and Technology, Trivandrum. He completed his BVSc and AH from Kerala Agriculture University in 2003 and Masters from University of Copenhagen, Denmark in 2011-14. He has worked as Research Associate at College of Vellini and as a veterinary surgeon in different divisions in Biomedical Technology Wing in Sri Chitra Tirunal Institute and now as Scientist E in Division of Laboratory Animal Science. In 2003, he has granted with Novo Nordisk scholarship to do master's project in University of Copenhagen. He has over 33 peer-reviewed publications in indexed journals and several book chapters. He has won the International Young Scientist Award by Japanese Association of Laboratory Animal Science at Kyoto, Japan in 2009. He was first rank holder in Animal House Supervisors Training Program organized by NIN Hyderabad in breeding, management, care and health monitoring aspects and yet more. With all respect, I humbly welcome Dr. Hari Krishnan sir to this webinar. Welcome, sir.
Thank you, Harita, for a much elaborate introduction given to me. And welcome all, good evening to everyone. So I hope I can deliver something which is useful for all of your careers in the coming years. And we will start right away. So this is regarding the role and scope of veterinarians in biomedical research. So the agenda is, let me just take 1 second to minimize the window. So the agenda is to discuss on roles and scope of veterinarians in biomedical research. And in roles, we will discuss in depth about broadly or in the details about the laboratory animal science field and the subject areas involved in the field and the legalities involved in India and abroad, and the institutions and association at the national and both the international levels. And in the scope section, we will deal about opportunities for education and training given to available at the national and the international level, and of course about scholarships which could be of interest to all of you, and finally about the employment opportunities available in the field. So all in a national and both international and national level as well. So the expected duration of the talk will be something like 60, maybe I will take a few minutes or maybe I take a few more minutes, I don't know. So regarding myself, Harita has given me a deep and detailed introduction. So in short, I got admission in to the Veterinary College Manuthi in 1997 and graduated out of the college in 2003 and did a few months of project as a research assistant in College of Agriculture, Vellini. And after that, in 2003, I joined Sri Chitra Tirunal Institute in a coronary stent evaluation program. So in pigs, so in pig models. So what is coronary stent evaluation? So that was the first time myself was coming to know about such a fascinating field is there, awaiting for us to give us jobs and prospects. So what is coronary stent evaluation? I will just mention you in a few moments. I'll spend a few moments to tell you about what it is all about. So this is the pig's heart, and these are the coronary arteries. So to give a detailed in-depth view, you can see that there are 3 major arteries in the heart of humans. So one is the right coronary artery, and then comes the left big branch, and left branch again it subdivides into two. One is the left anterior descending artery, the long one down the line, and one goes to the other side of the heart, which is the circumflex artery, left circumflex or LCX. So these are the three major coronary arteries present in the heart of humans as well as in pigs. So their vasculature, their branching is almost just the same as in humans. So we are picking up pigs as a model for studying coronary stents or deploy stents and angioplasty and things like that and related pathologies. So what we are doing is that in atherosclerotic plaques, so these coronary arteries, as you know, they supply blood to the heart muscles for it to be healthy and get nutrition. So when the blood supply to the heart muscles itself gets blocked with some atherosclerotic plaques or fat, then what we should do is we will do an angiography generally in human patients to see the extent, how extensive the block is or the plaques are. So if it is quite extensive, spreading to quite a few centimeters or something like that, the only option is to resect the artery. During that patch will be taken away to sew in another blood vessel from maybe autologous blood vessel, as we call them in human patients. The blood vessel from other part of the body will be taken and sewn. And for that, you have to stop the heart, and it is known as a cardiopulmonary bypass surgery. So apart from that, if it is a focal one, like this one, it is not quite extensive. You need to correct this area only. So if there are 2, 3, or 4 blocks in multiple areas, then it can be managed with something known as stents or balloon angioplasty. So the balloon it is guided with the use of a fluoroscope, which is an X-ray, again a live X-ray with contract dyes injected. So it passes this lesion area and it crosses the lesion area. It will have something like a refill, a crimped thing, a metallic thing over the balloon, which is known as a stent. So it is there, it is not inflated. So once it passes this area, the balloon gets inflated, which we can put some pressure to the catheter tip, which is outside, and then the stent will be deployed there to resuscitate the blood flow. So this is how the stent looks like. So there are many types of stents. There are pretty something known as bare metal stents and drug-eluting stents. So you have quite a lot of drugs-loaded stents, which what the drugs will do is that it again prevents the regrowth of the neo-intima of the blood vessels. So such a stent was evaluated. So before we can deploy that or before it reaches the human trials or in human patients, before it hits the market, you need to ensure about the efficacy as well as safety in a larger animal model, and we did it in pigs. So a Gujarati diamond cutter used the same technology of cutting laser. The laser which they use to cut diamonds are the same technology. They are actually diamond cutters who came into this field, and they used the same technology to cut stents, and they got CE mark, with which CE is certification of Europe. The biomedical devices require such certification for them to export to the European Union. So they got it with the data which we generated. So that was what I was doing maybe 17 years ago. So something happened. Reena, can you help? Something I can see on my screen here. Someone help? Something happened here.
Oh, sorry, sir. We will correct it. If you
okay.
Participants, kindly don't scribble on the board.
Thank you so much. So the areas worked in, I started with coronary stenting in pigs, as I was doing the anesthesia in pigs, and all those pigs we were exactly treating the same way how humans are treated for such a procedure, and they all came out and we evaluated the thing. And after 2006, so that was in 2003, and in 2006 I started to work with rodents and rabbits. And the current area of my work include facility management, all with rodents and rabbits, quality control, breeding, care and welfare, and creating surgical and non-surgical animal models for various research programs. And in particular, I'm interested, or my current hot topic is refinement of animal models. We'll come to all of it later during the presentation. So after a 6 years gap, I did my master's from University of Copenhagen, Denmark, in a part-time project which I did from 2010 to 14, and then a registered. I'm now currently I am registered for a PhD as well in the same university, and I am working in the refinement of spinal cord injury rat model. This is my web page. So this is the education which I got, and it was previously categorized as A, B, C, and D, and the D was meant for veterinary surgeons as a specialization. So the job in short is to create the diseases, to study the efficacy of therapies, or to look for signs of efficacy and safety of therapies in naive or healthy animals, to predict the outcome when human beings are treated. What happens to the human beings if they are treated with the candidate drug or biomedical device. So the therapies can either be drugs used in pharma research, but now I am working with biomedical devices or medical devices, as we saw in the case of coronary stent. There are heart valves, there could be blood bags, there are orthopedic implants, and lots of stuff. Any of the newly devised or developed candidate medical device or drug before reaching the market, we need to do the safety and efficacy in studies in either naive animals, which are healthy animals, or in disease models. So basically we need to create diseases in animal models or to inject them or to implant them with devices to see whether they pick up any diseases or symptoms of diseases or abnormalities, and detect that and convert those observations as data. So maybe we are just, or I am just doing the reverse of what we were trained from the veterinary college. So we were all taught, or we are all getting taught how to cure the diseases, whereas I am now doing just the reverse since 2003. So philosophically, how do I justify myself? I call it as utilitarianism. It is not me who am calling this. If you can go and visit this animal ethics dilemma.net, they tells about ethics in depth. So what ethics is, I will just use 1 minute or less than that to try to explain what ethics is. Ethics is different to you, to me, within me. The ethics which I had while I was a kid or I was in the college is not the ethics which I am having. We are all evolving. So as per the geography, a person in Africa would not be having the same ethical outlook of a person who is living in the Europe or in the Middle East or in India. So geography wise, within the person, experience wise, age wise, our ethical outlook differs. So, but there are a few points where we are all agreeing to particular ethical viewpoints, and those viewpoints are being described in the Animal Ethical Dilemma website. So you can go in later and sign in and create an account and see who you are, whether you are a contractarian or a utilitarian or a rights activist. There are no chance, or there are very feeble chance that a veterinarian or a scientist can be an animal rights activist, because animal rights activist view is that you cannot do any experiment, whatever the outcome may be. You tell that you have developed a cure for COVID. Tomorrow, you are not even the rights activists. Their view is that animals also are having equal rights. You cannot do even if you are telling that you have developed a COVID vaccine in maybe 5 months, they won't allow. But utilitarianism is something like a weighing balance. You are having two pans in a weighing balance, like like the statue which is in front of the judge. So on one side of the balance, he or she decides what harm we are inflicting to the animals, and in comparison to the other pan of the balance, which is weighing with the benefits caused to the human or to other animals. So more and more the benefit, that pan shall weigh more, and the balance shall weigh that side or tilt to that side. If it is tilting to the side of the benefits, then we are utilitarians, and we are telling that it is the benefits are more than harm, and we are for doing animal experiments. So that is who I think I am, but you can also check who you are. So, however, the standards are kept very high scientifically, morally, and legally. When we talk about laboratory animal science, there are three points to be taken care of. One is science. One is moral or ethics, which we just dealt with just now. And then is the legality. Whether you are a utilitarian or a contractarian or a whoever you are, you are bound to the national law or the regional law. So you are supposed to be abiding to that law on whatever your moral grounds could be. But scientifically speaking, why we have to follow the science is because we want to ensure that the data which we generate from these animals are authentic. So for that, we need to ensure the welfare. We are not supposed to do experimentation in compromised animals or welfare compromised animals, or the animals which are chronically stressed. Why animal experiments? I will just show you a few slides of examples why we are batting for animal experiments. So this is cocaine syndrome, a defect due to the ability of some cells in the body to repair the DNA that is getting transcribed. So the hereditary disease with ataxia, deafness, mental retardation, and fast aging. Transgenic mice are extensively being generated and being used for the research to discover the cause of this disease. And this is rheumatoid arthritis. So it can be a life-threatening disorder as well. The autoimmune disease that results in a chronic systemic inflammatory disorder that may affect many tissues and organs later on. And even now we have not totally unraveled why this happens and what exactly could be the cure. And even the diagnosis is a bit tricky regarding rheumatoid arthritis. And animal experiments are still ongoing to understand the mechanisms of both its cause and its cure. And we all think that meningococcal meningitis has a vaccine, but every year or periodically a new strain arrives, and we need to do repeated animal testing to ensure the production of efficient vaccines in meningococcal meningitis and many other diseases as well. And even for vaccines, when they are produced, batch-wise testing is there. For every batch, you are required to ensure regarding the efficacy as well as safety as per regulatory instructions or legalities involved, and then you have to, for the same invented vaccine, you have to do animal experiments repeatedly. So there are two types of groups which are telling that you cannot do animal experiments. We call them as animal rights activists, and there are other group the animal rights activists are also known as anti-vivisectionists. And here you can see a group of humans or a team telling that they are alive due to because of these animals tests. And it is calculated that maybe 20-25 years we have more of lifespan in comparison to 50 or 60 years ago, all owes to animal experimentation. Now we will see what laboratory animal science is. So laboratory animal science, somebody again is scrolling, so is drawing some lines on my cute rat. So can Anandu or someone tell? Yeah, thank you. So the multidisciplinary field that includes biology, pathobiology, all basic veterinary medicine and science fields. You name a field, it is there in laboratory animal science. Even statistics is there, surgery is there, you name the things. Even extension is there. Once you are done with the development, you need to go to the humans and tell or do surveys and even before the engineers or the biomedical scientists or researchers decide upon what biomedical device they need to produce in such a geographical area, they do surveys as well. So there are lots of field, and almost all the things or stuff which we have learned, pharmacology, toxicology, everything is here in this broad field of laboratory animal science. So the areas in specialization is again explained. We can basically classify laboratory animal science, or even laboratory animal medicine is again science. You can classify it in both the sides. So we basically, laboratory animal science or medicine can be classified as basic laboratory animal science and applied laboratory animal science. So there is a line somewhere here which broadly classifies them into two. And the first one is basic laboratory animal science. So this basic laboratory animal science is how we are going to house these animals, how we are going to care for these animals, how we are going to feed these animals, how we are going to breed these animals, what welfare, how are you ensuring welfare to the animals, whether the things which you are supplying to these animals are of perfect quality or not, which does not hamper with the biological functioning of these animals. So that area is known as basic laboratory animal science. And coming to applied laboratory animal science, the using of animals for testing hypotheses, that is applied research. For example, drug research and lots of other things. If you are using your animal for any of your animal experimentation goals or any kind of experimentation goals to generate data for something else, then you are doing an applied laboratory animal science. In other words, basic laboratory science is the quality of the animals, ensuring the quality of the animals as research tools. Whereas applied laboratory animal science is the development, validation, and application of animal models in animal experiments. So basic laboratory animal science include animal housing and environment, health, breeding, and genetic monitoring, health monitoring, genetic monitoring, breeding, genetics, everything, management, and quality control. So these are all the components of basic laboratory animal science. And how are the standards for these laboratory animals set? Are the standards properly maintained? Is there any scope for improvement? So to know this, you have to you have to think about how the animal as standards are tested. You have to know a bit about history of these animals, which we will come to the history of these animals a few slides later. The standards, we have seen that there are lots of standards mentioned in the slide. So what are all the standards? So there are American standards, there are European Union standards, and there are Indian standards. Basically, we use these three, or the combination of these three, to ensure the welfare of these animals, or to implement our basic laboratory animal science protocols or system procedures in place. So all these standards are free to be downloaded as PDF. So the guide for the care and use of laboratory animals, 8th edition, is for the American standard. So you cannot call it as a standard because it is not mandatory, but in the America, the legality works a bit differently. But the thing is that the guide offers everything which you require to run an animal facility, basically. And you can read the whole book as PDF. You can even print it. It is free for you to do that. And in the US, what they do is that they base these guidelines and they form so-called IACUCs, which are institutional animal care and use committees. They can decide what the rules are for that particular institution. But there will be a lot of people, veterinarians, socially aware people, and ethicists, biologists, everyone, a representation of scientists, and they together will decide upon their operating procedures, but they will definitely make use of the guide for the care of and use of laboratory animals as the base document. But the European Union, they have strong law set in. So this is known as new European Union directive. It came out in 2010 and got into action a few more years later. You would be able to download the English version of this. They got several European countries speak several languages, as you know, and they got the document available in each and every language, and it says in depth even the things like euthanasia and anesthesia, when to intervene, how to ensure pain, and all these things in detail you can see in the legal document. This is a legal document, whereas the guide for the care and use of laboratory animals has followed by the Americans. It is not a legal document, it is just a guideline. But this one they must follow. It is a must follow document. It is a legally binding document. And from an Indian perspective, you can find a PDF of a compendium, which is quite recently this thing. What is CPCSA? I will come to that a few slides later. But this is our standard, and if you could go deep inside this big book, you will be able to see the guidelines, which are, of course, maybe just the replica of the guide, the suggestions or the guidelines offered by the American and the European guidelines. You can also see in the Indian guidelines. So previously, the CPCSA was under the Ministry of Environment, Forest and Climate Change of Government of India, but whereas from this March or April, the second Modi government, they converted it to the Committee for the Purpose of Control and Supervision of Experiments on Animals, or CPCSA, is being transferred to the Ministry of Fisheries, Animal Husbandry and Dairying, and they rightly did that too. And this is, so now we know about, there are two basic classification in laboratory animal science, or the science is, the field is deeply divided or broadly divided into two, and this is basic laboratory animal science, and what the standards are to be followed or are available in the website. So all these standards, they make use of the same concept. So the laboratory animals, if you let it be a mouse or a rat or a guinea pig or a rabbit, or even larger animals like sheep or pigs or even dogs or monkeys, so we are rearing them in a controlled environment, right? So what we supply to these animals, they are being they are enjoying what we supply, and they don't have any access to seek for their choices. So there are environmental factors, chemical factors, microbial factors, and physical factors. If we can classify what we supply these animals into specific classes. And in the environmental classes, there are temperature, cage, food, and humidity, lighting, noise. So this could be physical as well, noise and pheromones, population of animals per cage, pathogens, ammonia content in the air or inside the cage, the bedding which we supply to these animals, miscellaneous chemicals like cleaning agents and things, or the spirit or surgical spirit which we use, insecticides which we spray to keep out the pests, the air which we supply, the water which we supply, the food which we supply, and the quality of and type of material of the cage, the transparency of the cage, the light emission, the noise. So everything has to go through a process known as quality control, which we have to do. So that is the basic job of a veterinarian, to ensure whether, or we have to first read and understand what are these factors and what are the values involved in all these, and we have to ensure the quality control before we supply these animals with all the supplies. So that is the basic job of the basic laboratory, or that is what the basic laboratory science is all about. Lab animals used globally. So whenever we talk about laboratory animals, we are most of the time, maybe 80 or 90% of the time, we are talking about rodents and rabbits. Why? Because 80% or 90%, 80% or even more is just mice and rats, and mice are more. So whenever we talk, most of the time, we just speak about laboratory animals, then we are knowingly or unknowingly, we are telling about or speaking about rats and mice. So we will discuss a bit about rodents here. So this is just to give you a broad idea of it is not for you to learn in depth what the rodents is or what their biology is, what exactly are the standards all about, but I'm trying to give you a broad picture of how it is dealt, how the basic laboratory animal science is being dealt with. So they are under the order Rodentia. You can see a mice here, a rat, a guinea pig, a hamster, and a gerbil, which we don't generally use gerbils, but in the European Union and in the America, in the US, they use it a lot. So these animals, basically, the history, we will talk about history. They were all wild animals. In Sanskrit, mush or mushik was the term used, is the term used for to denote these rats and mice, maybe mostly the mice. And from the mushik, the mice, or the word mus, M-U-S, mus came, evolved. So musculus is the scientific name of a domestic mice, and it shows that it could be somewhere in Asia. Some people claim that it is in India that these were originated. These animals were first seen or noticed, and when the Europeans came here to take out the spices and the granaries and everything in their containers or vessels of the ships, they as you know, they steal into everywhere. They even pest proofing is a misnomer. You cannot proof pest out of anything. They steal into everywhere and anywhere. So they also got into those ships, and they crossed the ocean to reach the Europe. And from the Europe, when the Americans or the Europeans found out the new world, which is known as America, they crossed the ocean again and to reach America. And in America in 1903 or somewhere around in the early beginning of 1900s, the first laboratory animal known as Wistar rat came into existence. How did they come? So as you know, the Indian rat has crossed 2 oceans over a period of time, reached America, and there are lots of pet breeders who were breeding rats. Why? Because they digs deep, makes deep pits in the ground, and they let loose all these rats which they were breeding, and they let loose their hounds to hunt them, and they were counting how many rats each of their hound or dogs were able to kill, and the one's hound which has killed the most number of rats wins the bet. So some kind of fancy pet breeding and fancying, betting, everything, and drinking and things were going on. And one person peculiarly noted a white animal, which was a mutant. So he identified a set of animals were not having any wild or dark skin color. So he identified all these animals and started to make a line breeding, and the result was the first laboratory rat. So a 100 years or 120 years ago, what happened in America? So you know now how the rats came into existence. The rats and the mice, they crossed the Asia through the ships, somehow reached America. They were all custom bred, and now they are coming back in flights, and it carries a huge price. For example, I brought some rats, maybe 24 rats cost me something like 2 lakh Indian rupees. That is in brief what the history is about the laboratory rats and mice. And these were the first type of cages which they were used to be in the laboratories. And from there, 120 years afterwards, you are seeing cages like this now. So their behavior and welfare, we should learn. So as you know, these are all wild animals, and the history of laboratory rats and mice, the behavior, we should learn a bit about the behavior in the wild, right? So we are designing new cages and all. We should know how the animals were behaving in the wild. So these rats and mice, they are extremely fearful. We call it as neophobia. They are even fearful to new types of food in the wild. If you place some new objects, they won't come near the new objects. Why? Because they are all prey species, and they fear that somebody will be trapping them, somebody will be baiting them using poison. That is exactly why they are showing aversive behavior against new kinds of feed, new kinds of objects, because they fear that it could be a trap, all set by cunning humans and all. So this is their history, and they show properties like thigmotaxy, where they prefer to go through or walk through, avoid open spaces, and walks through just nearby to the walls, to ensure that no predator is easily able to locate them and fly down and catch them and eat them. So thigmotaxy. So these are some properties, and they are aversive of bright light. Why? Because they are again a prey species. They avoid they don't prefer white light because to avoid predators. So these are some of these characters based on which we should know why. Because we have to design cages. So we have to build cages which should suit their requirement. Without understanding their basic behavior, we cannot do that. So the basic laboratory animal science does a lot of research in this area, these areas which could replicate the conditions which they were enjoying in the wild, at the same time not compromising the experimental setup and easiness of handling and cleaning their cages and all. So you can see such a cage, it transmits almost all the light, 100% visible light. So the animals must be undergoing some kind of stress if they are housed in such cages. So this is what our research is all about from the basic laboratory animal scientist point of view. The temperature, relative humidity, air changes per hour, everything is under control. So it is our duty as a basic laboratory animal scientist or veterinarian, of course, to understand the standards and to ensure that the standards are well maintained or not. And so the basic laboratory animal science routine jobs include the maintenance of statutory and regulatory compliance. So what are the statutory obligations as an Indian laboratory animal vet, which we are obliged to? So we have to follow the CPCSA rules and regulations. We will come to CPCSA in quite a few slides away. And from the regulatory compliance, there are different quality platforms, like ISO based platforms are there, OECD guidelines are there. If you are a drug manufacturer, you follow CDSCO or something. If you are a calibrator, you follow so and so. If you are a biomedical device evaluator or researcher, then you follow ISO. So there are lots of regulatory compliance which we have to undergo through. So you have to follow, as per which institution you are working, you have to follow these set of guidelines or regulatory documents. And you have, of course, irrespective of where you work, you are supposed to follow the CPCSA guidelines, provided you are working within Indian Union. And here you can see a AAALAC. If you can go, it is an Association for Laboratory Animal Care of American origin, which ensures that we offer high quality welfare standards and research standards to these animals. So if you got an ALAC stamp accredited to your certificates, it gives you much value, and it requires some money also to keep in place ALAC accreditation and these kind of regulatory compliances as well. So as part of a routine job, we are supposed to keep all these accreditations as per your institutional norms in place. And the routine jobs includes breeding, health management, ensuring supplies like feed, ensuring bedding materials, ensuring drugs and things like that, and quality control of all supplies. Some will have to undergo autoclave, and then again you have to evaluate the efficacy of efficiency of the autoclave using some strips and things. And some materials you cannot autoclave, will have to go through a UV chamber, or some will have to go through a chemical dip. If you are a human being walking into the animal facility, you will have to undergo through a health checkup and a shower and a PPE change. So, and if you are bringing in animals from outside, you have to make them undergo through quarantine, and during the quarantine procedure, you will have to decide whether these animals are lodging some subclinical diseases or not, because all these rodents, as you know, they are culprits of all the major outbreaks like plague and lots of other things as well. Hand-tan virus, and lots of viruses are there, which without showing symptoms, these animals will be having. So for that, you have to ensure the quarantine health procedure is properly followed or not. And then you as a veterinarian in charge of a basic laboratory animal science unit or an animal facility, you are supposed to write operating protocols on various actions which you are performing, like feeding. You should have a protocol. And you are supposed to train the staff, your subordinates, newly joined staff, and training and retraining and periodic getting yourself trained is another requirement. And then planning and supplying of animals for research groups, and most importantly, ensuring animal welfare is our job. So how the standards are set, how are these standards set? So we were somewhere around discussing how the standards are set. While we again rewind what I had been telling a few moments ago, we were discussing about the history of these animals, how these animals behave in the wild. Even after 125 years of captivity, still these animals possess all the instincts of the wild rodent. So there are proof that animals still have their wild instincts intact in them. So we need to know how these animals were behaving in the wild, so that you know about the standards and how the standards are set. And we should also be competent enough to know that or assess that, is there any scope for improvement in your housing standards. So the research in basic laboratory animal science is there. So apart from a routine job, as I enlisted here, it also includes a research component, based upon which we assess that whether the standards are adequately set or not, and is there any scope for improvement or not. So the history of laboratory animals and rats and mice, you know a bit now, and the behavior in the wild, and what do these animals need in captivity. As I told you, these animals are all prey animals, and they spend a lot of time to escape from the predators. That is why they live in deep burrows, subterranean. We call them subterranean burrows. They burrow a lot. They have ever-growing incisors, which helps them to burrow and even chew away wood and stuff. And they doesn't love bright light, so they burrow deep inside the subterranean terrain and breed there. They live in social colonies of one male mice, many females, and many litters, and so on. In the wild, they also do a lot of grooming themselves, like the cats are doing. They lick a lot of themselves and tries to, if they are healthy, they will be looking very nice and clean. and they have to find a source of water is very important for these animals in the wild. So they actually travels a lot per day and finds or identifies a place from where they can find water and also feed. And these mice in particular, these animals, both animals, but mice in particular are very good nest builders. They build nests for their young ones, and they build very complex nests. If you supply them with nesting material, which are shreddable paper strips or hay or straw, they will show you how to make very complex nests, which is really wonderful to see. So they in the wild, the point of this discussion is that in the wild they use lots and lots of time to escape from the predators, to make burrows, to make nests, to find feed, to groom themselves, to breed, things like that. But in the captivity, within the four walls of such a transparent cage, what do you expect them to do with all the ready-made food? All the 100% of the time they are being provided with water, adequate bedding. They don't have to fetch for bedding material. They don't have to run around and escape from their predators. They have well-planned life cycles. Everything is standard. So what happens? What do these animals need in captivity? So they turn into aberrant behaviors, because there is a lot of free time or leisure time to these animals, because what they were doing or performing in the wild, they doesn't need to do that anymore within the captivity or within the four walls and the lid of a cage. We think that is comfort, but actually these animals can start self-mutilating, fighting behavior. They expect we can expect fighting in male mice, aberrant behavior, which I have a video which I will show you later on. So self-mutilation, turning aggressive, and pica behavior. So lots of aberrant behavior can happen as well. So we need to assess what is going on within the four walls of the cage, and we should be able to suggest changes to our laboratory animal colonies from time to time. So that is also our job, or that is also a part of the research in basic laboratory animal science, and it is highly evolved. The research on housing condition, environment provided. So a simple thing is that we can provide them with preference tests. We can offer them with a range of variable things to choose from, maybe 5 types of feed, and see which feed they prefer. So things like that are there. But there is an example which I show you. I'm going to discuss to explain to you what research in basic laboratory animal science is all about broadly. So the temperature range, we can take the case of temperature range for housing. So basically the temperature range which the housing is done in laboratory animals is the air conditioner's temperature, which we set inside the animal facility or inside the animal rooms, is between 19 and 21. In some places it will be 22 as per the standards which you are following. So we think that it is the standard says so, and these animals are really comfortable in those standards. But recent research says that the thermoneutral zone of mice is 26 degrees Celsius to 34 degrees Celsius, and below 30 degrees, the mice could undergo through thermal stress or cold stress rather. The nesting material, which is a type of bedding material, which like blankets, which we are having if we are putting the air conditioners on. These animals are also being provided with nesting materials in good animal facilities, and especially in the case of nude mice, which doesn't have any hair on them. To young animals, we have to provide, because they again doesn't have any hair on them. And as an enrichment to the adult mice, we are supposed to provide them with nesting material. So this is the case. the 21 degrees Celsius of temperature which we are providing to these animals in the room, whereas they would, their biology shows that it is something around 30 degrees Celsius which they require. So what reality, why are we providing 21? So some studies were set up and like the ones, like what I told as the preference test. So such a test was being conducted, and what about the animal cages in 3 temperature ranges were offered, and these animals had freedom to move and pick up whichever temperature, whichever cage with whatever temperature they would like to reside in. So 20 degrees Celsius, 25 and 30 degrees Celsius were the 3 temperature ranges with bedding only was provided. And the mice which are nocturnal and active in the dark phase, when it is active, more body heat is created. When it is inactive, less body heat is created. And next question, do the preferences of mice correlate with the level of activity? So all these factors were considered during the setting up of this experiment. We considered that the animals are active during the dark phase, so many readings were taken during the dark phase, many during the light phase as well, to see whether some differences are there during the inactive phase, which is the light phase. And the results, if you could see the results during the active phase, which is the night phase or the Skotov phase, they didn't show any difference because they were all active. Expectedly, their body temperature ranges while they are active were high, and they didn't mind even if the temperature was 22 degrees or 19 degrees Celsius. They were already warm. And during the inactive phase, they preferred 30 degrees Celsius. And the standard says it is 19 to 21 degrees Celsius. So there was a significant difference between the 3 groups of mice. And during the maintenance phase, the significance was not that drastic as we could see that from the inactive phase, but again, a difference was there during the maintenance phase as well. So these all suggest why the standard. So we are questioning back the standards. Why did the standards fix 22 degrees Celsius? Again, the regulatory bodies or the standard fixing organizations, they will have again some sort of arguments to make, like in the Europe or in the America where most of the animal studies are being conducted, maybe at least during the period during these standards were framed. So they were considering that their winters were minus 20, minus 30, minus 10, and then to bring it up to 22 was fair enough, good, rather than bringing it up to 30 degrees Celsius to save a lot of energy, but still that made a huge difference in the housing of these animals. That is why they compromised for a 22 degrees Celsius. Another argument could be that in research capabilities or research facilities, we are intensively housing the mice. Of course, we are basing everything onto the floor space requirements, the standards asks us to do, but still we are housing animals to the maximum level wherever we are thinking about economy, where we are thinking about efficiency of the facility, everything. When these factors are considered, we are housing the animals in groups, and the groups can generate heat, and that is why they are sticking on to 22 degrees Celsius. So this why I told all these things. This is not to teach you that this is a research which is going on and this research is important, rather than I wanted you to think that or to see that this is the kind of research involved in basic laboratory animal science between regulators, between housing bodies, and there are lots of funding agencies to find out the welfare. And this is welfare. Basically, if the animals, which the standards say, when the standard says it is 22 degrees Celsius, and if you are housing them in when the biology says it is 30 degrees Celsius that these mice prefers, and the experiment says it is 30 degrees Celsius that the mice prefers, and the regulatory bodies and the guidelines of the standards and the legal documents say it is 22 degrees Celsius or 19, there is a compromise of welfare involved. So the basic laboratory animal scientists are deeply concerned over these kind of welfare issues, and there are agencies which funds us to work more to find what are all the welfare requirements and what didn't be addressed so far, even after 125 years after domestication and making these animals as a laboratory research animal from the wild. Another small example is the presence of an endogenous biological clock. So this experiment will tell you that the reporting of the findings, all the conditions which you were using are really important. So these animals, as I told you, they are all kept in captivity, bred in captivity. They don't have any access to the sunlight and no access to the moonlight. Maybe they don't have any access to the rain which is falling, the wind which is blowing out. Why? Because they are all under barrier maintained conditions, and you are changing your PPE to change your dress before you enter the animal facility. You are changing your footwear. You're showering yourself to take out all the smell and dust particles to give them any clue of what is happening outside. So the barrier facilities are all over the world, and it is upcoming in India as well. There are several good barrier facilities, and SCTI MSD is also in the process of revamping our animal facility and upgrading it into a barrier maintained facility. So the barrier facilities doesn't, the basic thing is that the barrier is the microbiological barrier or the physical barrier between the external environment and the internal environment. So the laboratory rodents or rabbits, they doesn't have any clue what is happening out. But still, an experiment was done in 2006, and in the US, I think, by a group in US, and they collected serum corticosterone concentration to get a clue on their stress levels, and they found that the animals were significantly different, having differences in stress levels during spring and autumn. So this is a proof that these animals are able to find some cues or some clues that's what is happening outside, even if you shut out everything. And this is, more importantly, this basic laboratory animal scientific research which was done is applicable to the applied area of biomedical research, because two groups are doing the same experiments spread over 2 seasons, say spring and autumn, and they doesn't know that such a factor exists, and they were comparing their results, and they found that those they similarly followed their materials and methods, yet they got different results. They came out with different results, and they doesn't have any clue, and they blame it all about repeatability or trustworthiness of research groups, between research groups, and ultimately it is the research data which should be really authentic which gets suffered. So by generating data like this, we are enlightening the applied biomedical researchers or even the basic laboratory animal scientists that we shall be cautious in while reporting, and there could be confounding variables like this present in your research. So now I hope that you have got some idea about the basic laboratory animal science field, and now you can see what is there in applied laboratory animal science field. Drug and vaccine research, I'm not going to discuss much about that. You know what it is. Medical implants like stents and valves, which goes inside the human body or inside the animal's bodies, like orthopedic implants, dental implants, and things like that. And genetics, of course, we saw Cockayne syndrome. So if we need to know more about the pathobiology there, the DNA structure and what happens exactly to the DNA and what cure, where to intervene, etc. You need a lot of animals in genetics. And in education, all the educational institutes, including the COAS Manuti, has got an animal facility for promoting education, doing their own research projects and all. And medical products like blood bags, here you have seen medical implants which goes inside the body, but outside the body there are lots of medical devices like heart lung machine used for cardiopulmonary bypass, oxygenators which are used during the cardiopulmonary bypass, blood bags, simple equipment like even like syringes and things like that. When you develop that, when you batch wise ensure their authenticity, you need to do animal research or research using animals. And that all comes under applied field of laboratory animal science. And the space, and even in space and different, you might have heard about a dog, Leica, which was sent to the moon. Maybe lots of rodents, and they study a lot. We don't know the exact details because they are a bit confidential, but still DRDO and space research uses a lot of research animals. And in human behavior and neurology, so you have to learn about human behavior. You can use animals because every behavior can be linked back to the neural mechanisms, to the brain, to the spinal cord, to the neural systems, to the catecholamines or neurotransmitters, things like that, including Alzheimer's, including schizophrenia. So you cannot go on and dissect a human being and test all these hypotheses of yours. For that, you need, there are schizophrenic mice model, there is Alzheimer's rat model. So there are fascinating models available. And we as laboratory animal scientists in applied laboratory animal science field are supposed to know the wideness or the breadth of all these the implications of all, or the applications of all these things, and how to create as per in which field you are getting focused or being posted. So human behavior and neurology, basic research, there are lots of basic research happening, and toxicology of drugs and chemicals. So of drugs and drug toxicology is an industry in India. Maybe Ahmedabad, Bangalore are the hubs, and also Hyderabad could be the hubs of these drug industry. There are lots of drug manufacturing companies involved in drug research. So what is our role in applied laboratory animal science field? Apart from doing research in all these fields which were mentioned, to do the welfare assessments in the animals which are undergoing these research in all the experiments, and your presence as a veterinarian during the experiments. So who are in charge of these facilities in India, or all these let it be basic or let it be applied areas of laboratory animal science, who are in charge? So upon a few years ago, maybe 10-15 years ago, they were fully run by, or maybe 80 or 90% of them were non-vets or the PhDs in zoology or just MSCs and MSCs in zoology, people who were running the show. But there were many veterinarian stalwarts also were the part of our rich history in the field as well. So we were not given that much of a representation in the field of laboratory animal research until very recent past. So veterinarians in basic laboratory animal science, we will see what do we lack or what are all our weaknesses actually. So there, as far as I got past in 2003, as I told in my third or fourth slide, but still during that time at least, we didn't have any rodent biology and diseases, and experimental surgery was also unfortunately not the part of our curriculum. And I don't know about the scenario now, but I heard that a few bit more chapters are being added in pathology or those areas to teach you a bit on laboratory animal diseases. But the adequacy of it, I'm not really sure about the adequacy of the syllabus yet. And higher education in this specialty for vets is not yet here in India, unfortunately. That's why, unfortunately, I had to, fortunately or unfortunately, I had to go to Denmark to do that. So the training centers of quality are only a few. So there are a few training centers from maybe since the past 25, 30 or 40 years. There are training centers, but only a handful. And of course, the knowledge is not among the veterinary community. They are not really popular. Even many of the working vets in India are lacking in exposure and in funds. Why in funds? So even if there are lots of veterinarians being posted nowadays in all these research institutions, I'm not telling about my institute because the institutes like my institute were really keen to train people like me and my colleagues to take it to the next level. But there are several institutes who are not training their veterinarians properly to gain knowledge, as well as the animal facilities are not channelized with adequate funds for them to get trained, as well as for them to run their research projects, and of course not enough funds for the upkeep of the facility as well. So we are all weak in these areas. What do we have? Our strengths. So we have a really good academic background because you all are well versed in surgical trainings and skills. You have a strong background and several courses of pharmacology, surgery, anesthesia, anatomy, everything. And with a few weeks of training, you could do wonders, unlike the MSCs or the PhDs can be doing. And the institutes and the emerging institutes, especially from the private and of course in the public as well, are increasingly understanding the importance and utility and the trainability and the ease of trainability and the pace where they can pick up things to a large extent nowadays. And now, one of the strengths which we are having is that CPCSA has made it mandatory, that very recently they made it a mandate that a full-time veterinarian is a must, or he or she must be posted in every institution which is having an animal facility. So this is the order which the CPCSA gave out in February 2019, which is just over an year from now ago, and it reads. I will read out, because if you cannot read that, I will read out. But and in case you are not able to follow any of these slides or websites or anything, don't worry about that much, because many of you might be viewing this through your mobile phones. I will be circulating through Anandu and the organizers the PDF of the entire presentation, so that you can scroll around and see the websites and in depth and go to those websites, and they are all free to be accessed, freely accessible websites which I have posted here, which you can do later. So this reads: it has been observed that the CPCSA that part-time veterinarians are not able to regularly visit the animal house facilities and are not able to provide 24 hours veterinary care to the animals as per the guidelines of CPCSA. Therefore, all establishments getting registered with CPCSA should have a permanent full-time veterinarian for ensuring the proper care and well-being of laboratory animals. So that is the legal document issued by the CPCSA very recently, maybe 1 year, and you should be really keen enough to follow it up with VCI and CPCSA also. So if you can go to this website of CPCSA, you can see the phone numbers and everything here. And if you scroll down this page, you can see the list of establishments registered with CPCSA. And if you click there, you will be able to find the PDF document of the recently registered or the current registered number of institutions present in the country. And then you can use your resources to find whether they have posted a veterinarian or not. And as per CPCSA, it is a mandate now. And secondly, you can also become a CPCSA nominee. So who is a nominee? So I have told about CPCSA, and CPCSA is the statutory body to which each and every research organization shall register with. If you are a breeder who breeds animals or rodents or dogs or monkeys or whatever it may be for the use for experimental purpose, which could be another organization, even then you should be registered with CPCSA, which means all the registered organizations which are entitled to do experimentation, education, or breeding for experimentation are all registered already under CPCSA. And CPCSA will post an IAAC, which is known as an Institutional Animal Ethics Committee, into each institute to see their operation and to see their protocols and see their compliance with the CPCSA guidelines. And if you are a CPCSA nominee, you will be posted as an IAAC nominee to any one of these local institutes, maybe quite near to your residence or office or to your college. And each and every graduate can apply to become a CPCSA nominee. So how to apply? So they actually conduct occasional 5-day training, 5 to 6 days. It is 4 or 3 or 4 or 5 days training which they are conducting, most probably in Delhi, New Delhi. You will have to bear the charges to reach New Delhi and to undergo this 5 days training program which is offered by the Ministry of, by CPCSA. So, and then you can become a CPCSA nominee. Usually the mail for, calling for CPCSA trainings is being circulated to institutes like this to send their candidates as nominees. But you can either keep in touch with some of the nominees which you know whether they have received such a letter from CPCSA recently or not, and can go in directly and apply on your own, or you can also call, make use of the number and contact the mail and write to CPCSA and ask when the next training is and can join that and become a CPCSA nominee. So CPCSA nominees, basically that what is their job is to visit the assigned institutes which is assigned by the CPCSA and take part in their proceedings. Like they conduct IAAC meetings as a mandate every year, twice or thrice per day, and as per the number of experiments which each institute is handling, the periodicity of the frequency increases between the meetings. And also you have to inspect their annual facility each time you visit, and you have to write to a report to CPCSA regarding that. And you will be paid with an honorarium of 5,000 rupees for each visit, plus your TA and DA as per central government norms. Some skill sets are required in experiments and welfare assessments. So the pain, so you cannot claim yourself as an expert, or just because you are a veterinarian that you have the license to treat laboratory animals, because it is our moral obligation to know more about the species, and each species has their own precipitation of pain, expression of stress, and behavior on a whole differs between species, and you cannot be anthropomorphically say that you know everything. So that you are supposed or advised to undergo some kind of training or education. So you are supposed to focus on the pain assessment in rodents, stress assessment in rodents, negotiation of pain and minimizing stress, experimental model creations like surgical and non-surgical models, and many more. And how can you acquire this? We'll deal with these in detail. Education and or training. So I'll tell you in a next few slides about further education. You know that I did my PhilASA category D, and at that time it was categories which were mentioned as PhilASA, and D was the top level category they were offering for a master's level, to create or to groom specialists in the field of among veterinary surgeons in laboratory animal science. But now later on they have modified their guidelines, and now it comes as categories A, functions A, B, C, and D. So what are these functions? So there you can go to the PhilASA website and read more about functions, but that is how it comes now. And specialized education for veterinarians is still present in the European Union and all online. So this is one such program which I did. So I had some contact classes. I had to go there quite a number of times. I had to undergo some practical and examinations there, Viva, OC, everything there. But they have, over a period of time, modified their curriculum, and it is now all online. So if you can go to this website and see the Lab Vet Europe program, it says or explains how you can become a designated veterinarian in European Union, but still you have to undergo some more licensing test if you plan to do practice in European Union. So I am a coordinator. If you require more details on this course, I can give you. So ICLAS regarding scholarships for this course. So there are a few handful of scholarships available for this. So you can go to the ICLAS website, International Council for Laboratory Animals Science. They offer a scholarship program for veterinarians in laboratory animal science and medicine. And they have told that they specifically offer these programs to meet the criteria, have agreed to participate in the scholarship program, and specifically the Danish program features there. So you can see a Spanish program and a German version of the program is there. So it is a full, not an online. As per my knowledge, you have to go there and spend your money to stay there and find scholarships, maybe to learn more from Aachen University of Germany. But in Denmark, it is totally online. You can go to this website and see and also apply for an ICLAS scholarship to participate for the course. There is a Laboratory Animal Science Limited, which is a registered English or British charity organization working in the field for the welfare of laboratory animals. They have a journal as well. So the Laboratory Animals Limited, if you go to their page and click on the funding area, it will show individual funding. So if you click on the individual funding, then there are courses, the guidelines on how to apply for funding comes, and you can see that the courses intended to which you are going to apply for must be accredited or recognized by PhilASA or any other competent authority. And the Danish course is also a PhilASA accredited one, to which you can apply a funding for an individual funding and try your luck. So there is UFA grants and awards. The UFA is Universities Federation for Animal Welfare, again an English charity society. So they offer a wide range of funds, but mostly, to my knowledge, once you start working or once you have completed your education, or once you have reached your education to the final semester or something like that, in the laboratory animal science area, you'll find it more easy to find funds from UFA. But if you are a beginner, like just a fresh graduate or so, Laboratory Animals Limited can be a good option, and so shall be the ICLAS for you to seek for funds. So there are these are the European trainings. There are American as well, but to my knowledge, it is really hard to access American trainings unless you clear a GRE and go directly for a PhD or an MS PhD. So Indian trainings are there. So this one is the best one which you can find in India. It costs you something like 25,000 rupees, or and the stay and the train fare to Madras Veterinary College in Chennai. But the thing is that it is again FELASA accredited. So the only one course which Federation of European Laboratory Animal Science Associations accredits outside the region, the European Union, is this course. So we are proud to be a part of FELASA accredited training course. Usually it falls by the Onam, just after the Onam time and all. So you can see this year, definitely I don't think that it is going to happen this year. But again, you can get a FELASA functions A, B, C, D and D accreditation from a 2-week or 10 days long course from Madras Veterinary College. So it can impart to you, it can exponentially bring in expertise and training to you, your skills. It can definitely be an asset if you wish to work in the field of laboratory animal science. This training can be. So there is training in Kerala, Trivandrum as well. Sri Chitra Thirunal Institute, my institute, is actually offering, I am offering a training. Usually it comes every year, twice, in January and in June, or in December and by mid-year. So every year it happens, but since the last year we have an interruption because of, I told that we are bringing in or setting up a barrier maintained facility. We are upgrading the barriers and revamping the facilities, owing to which we were not able to conduct it last time. But as soon as the COVID is over, and as soon as we are entering into the almost to the final phase of building or revamping the facility. So as soon as the COVID is over and everything is over, hopefully we will be again resuming that, and it is really cheap. And it is for 6 days, and if you are lucky, we can book an accommodation for you a cheap accommodation for you in our guest house. It costs only 3,500 rupees for 6 days, and we are also offering 2 teas and working lunches for all the 6 days as well. But this is a basic level training. Unlike I have told that about the FELASA accredited functions A, B, C, and D level training for 10 days by multiple faculty from all over India participates here to impart training in this course. Whereas this is a basic training which can just induce you to the field of laboratory animal science by shooing away your fear to handle the laboratory animals, to teach you the basics about the identification of pain and welfare, making welfare assessments in laboratory animals, collecting blood and body fluids, and administering drugs through various routes, anesthesia, and basics of surgery and things like that, and legalities involved like CPCSA and ethics, of course, in depth. So the announcement will come here, or you can write to me maybe by next year beginning, if the COVID scenario is gone. So train yourself for free. Meanwhile, while the COVID is still around, you can try this website. It's a wonderful website, NC3Rs, National Center for Replacement, Refinement, and Reduction of Animals in Research, and you can see e-learning resources here. And when you click on the e-learning resources, you can see videos like this. So I'm just playing this video for you to see. So this video is just an example of the video what the NC3Rs are offering. So it shows how the healthy mice looks like, how actively they are during within the cages without disturbing them, how you can observe them. So these are all the signs of activity where they climb, they actively feed, they walk through the beam, they are running around and playing with each other. And this is another example of a group of rats which are also really healthy. The shiny eyes, the well-kept coat, a group of healthy rabbits, again, which are healthy. So the rabbits actually, usually they are shy, and if you enter the rabbit or guinea pig room, they won't move at all. So you use video cameras to shoot them. Like you can see this guinea pig, no one is around, so it does their activity comfortably. Again, another video of mice which are all healthy. So this video, these kinds, and again you can see some peculiarity in these mice. It doesn't look that healthy because their skin coat is ruffled. And again, this is how you can handle mice without even touching them and without them injuring them, and at the same time you have all the access to see their examine them in detail. So this is another video again. Another video again showing you how the healthy animals looks like and how the not very much healthy animals will look like. So here you can see a group of mice which are actively out. Here again, mice are there, they are not coming out, they are hiding themselves. And when you open the cage of the active mice, you can see their coat, their activity, they are very highly active, their eyes, they look really healthy, and they move around, and they are there. Whereas the other cage where the animals were hiding, you can see the hunched posture, the ruffled hair, they are hiding, they are not that much active, they walk quite absurdly, and you can see this animal has lost some hair there, it has some kind of itching, and it is not at all moving at all, not moving at all, and it is a bit more obese as well. So it falls, it is not that normal, it doesn't look that normal. This is a normal guinea pig, but its walk, while it was walking, it had a slight this thing. So again, animals, you can see this animal is undergoing seizures, momentary seizures. So it is you can watch, they have undergone some brain implantation, skull implantation, and you can see sudden seizures which could all be abnormalities in these animals. So you can see stereotypic movement. It is not abnormal. If I rewind, I will show you again. You can observe this animal. It is repeatedly doing the same kind of meaningless movement which is termed as stereotypic behavior, and it is also making several somersaults, like in circus several times around, again, which is not normal. It is known as looping behavior in mice, and it is not normal. Sorry. So you can watch that abnormality in the behavior now. The stereotypic behavior going in rounds again and again, and looping, again going in round and looping. So kind of stereotypic behavior. So it is not normal. It all shows that these animals are not normal, and it suffers from reduced welfare. So this tries to eat the feed, but it is just chewing the bars repeatedly. So bar biting, it is known as, again, another expression of a stereotypic behavior, another abnormal behavior. So again, another kind of abnormal behavior, it just do what we call it as rearing behavior and keeps on like that. So these kind of videos you are able to access from that website which I showed as NC3Rs, and you will be able to answer quizzes as well later on. So there are videos on assessment of health which I'm showing since the past. This is the third video on health. You can notice that these animals are not healthy because they show a ruffled kind of skin coat, which is not normal. We already discussed that these animals, like cats, grooms themselves and keep their body really glossy and clean, but whereas this looks ruffled and it is not normal. So if you click on the next slide, you will be asked a question from which you need to select the correct answer, after which you will be prompted for more and more questions. And maybe if you spend 90 minutes, you complete a European Union module on health. And there are modules on anesthesia as well. So here is another example on anesthesia. So the example shows, the question is, would this mouse be a good candidate for anesthesia? So you should view the video and answer, so that you will be told the right answer. So this is a mouse. So it looks pretty healthy to me. So it is having a good skin coat and hair coat, and its eyes are bright and shiny, and it is quite explorative in its behavior, quite active within the cage. So for me, it is a good candidate for anesthesia. So I will pick that answer, and you can see the next video here. This mouse actually it shows some lost hair there due to a thing known as barbering, and its hair coat is not that clean or well groomed, and it is not that active. It keeps on there, and it seems like the animal is a bit anxious, and which renders it the answer to be the answer is that the animal does not appear suitable for anesthesia. It has a ruffled fur coat, probably indicating the lack of grooming, and its eyes are half closed and perhaps slightly sunken. It has pale ears and tail and is not active. So again, another example of how the animals respire during anesthesia. So this is the module on anesthesia. So it shows you how to anesthetize the animal, how to drape the animal, and once draped, you cannot see, because it is a tiny animal, and you cannot see whether it is respiring or not. So you should employ something known as, so something known as transponders or sensors for you to see their vital statistics like respiratory rate, cardiac function, heart rate, and body temperature. So which is also available in the market. So it will train you. These videos will really train you like anything. And it shows how to assess the respiration, whether the respiration is adequate or not. So this looks fine for me, but this looks a bit labored for me. So in this case, either you should ventilate the animal using an endotracheal tube, you should endotracheally intubate the animal. This looks fine for me, this guinea pig, the respiration in this guinea pig. So this is a bit shallow and might require some oxygenation. So like that, you can get trained yourself using all these videos and all these websites. And there are more websites like AHWLA. Again, you get a set of videos how to gastrically gavage animals, the procedures, every procedures you can see and learn. So now you are done with training and scholarships, and what about jobs? That's what you would be asking me right now. I can hear that. So there are lots of opportunities, and get notifications at the right time is the key. So there are many things happening around you, and for you it is important for you to leap to this field if you are looking forward really for getting some job opportunities in here. You are supposed to know the vacancies and recruitment and things like that at the right time. So you can, I am inducing you to LASA India, Laboratory Animal Scientists Association of India. Even if we have termed it Laboratory Animal Scientists Association in 2000, way back in 2004 or 2005, while I joined. I joined as soon as the organization was formed, the association was formed, and there were lots of scientists, as we were discussing, there were lots of PhDs and zoologists and everywhere who were there, and in the executive committees and the office bearers were maybe 50% or even more of non-vets. But now the scenario has changed, and there are lots and lots of vets there, and almost we are phasing out actually. I hope that there are no non-vets included by Anandu in the this thing in the group, so that I don't get beating when I walk out of the webinar. So slowly we are phasing out the MScs and biologists and other PhDs, and slowly employing veterinarians in the field, and slowly motivating these veterinarians to the field and to take up more jobs, become more CPCSA members and all. So LASA is driven now by 100% a veterinary team, and we got a website here as LASA. If you type LASA and Laboratory Animal Scientists Association of India, you can see the website, and there are areas where you can click here for viewing the membership criteria and all. You are, of course, you can become member as a student. It will cost you 500 rupees if you are applying now. If you have graduated already as a national member, it requires you to, or it costs you 3,000 rupees. And every detail is there in the Laboratory Animal Scientists Association. And what is special about this group is that we got 2 or 3 WhatsApp groups where members share their job vacancies. Like basically, there will be lots of BVSC positions opening up. There will be lots of pathologist positions opening up, and there will be positions where any vet can apply, or educational opportunities also showing up in these these WhatsApp groups. So all over India is covered. So there are lots of members from all over India. So there are lots of jobs in Kerala. Actually, not quite a lot, quite a handful. Usually the jobs are in industrial states, mainly like Gujarat, Delhi, Andhra Pradesh, and Karnataka, and even in Tamil Nadu, which is there. But in Kerala, we have got we don't unfortunately have many industries thriving very well in Kerala, and we don't have many private players here. But there are few institutions, mostly governmental, and recruitments occasionally happen, but not very frequently. And they happen on a contract basis as well when they announce. If there are any veterinary pathologists, I have a news for you that I heard that there is a soon upcoming call for a veterinary pathologist at our institute. And you can go to our institute's website, sctimst.ac.in, and then you can go to the recruitment tab, and you can see the enlisted recruitments. So I advise you to keep an eye here, because there are not a lot of opportunities opening up here, but there is one upcoming, and there will be occasionally 1 or 2 for BVSC candidates as well. So many medical colleges are there. There are many unraveled spaces which we will have to focus upon now, mainly because of the rule which we came across with quite a few slides ago, that CPCSA has made it mandatory to post vets in all, as a mandate in all the registered institutions where an animal facility is there. And there are many medical colleges, even in Kerala. There are many private medical colleges as well as pharma colleges are having research institutes, like ICER Trivandrum is having animal facilities as well. So there are lots of upcoming vacancies which I can see as well in Kerala. So you should care about what we have discussed a bit ago, that it is a year ago that it came, and what the medical colleges and basically the medical colleges, they have not, and the RCC Trivandrum, they don't have a full-time vet yet. Why? Because they are telling that they require more time to make their amendments and job positions to create, for them to create as per UGC, this thing to create a new post. They require time, and they are buying time from CPCSEA right at the moment. So we have to follow it up with VCI and with CPCSEA. So working abroad, now I will talk a bit about working abroad as a lab animal vet. So it is easy for you if you are trained and if you have some experience in India. You can easily go to find a job as a scientist in the US or in the European Union or even in the Middle East. But in the Middle East, the number of research organizations are not that much, but there are educational and research organizations in the Middle East as well, where you can apply as a lab animal vet in the Middle East, but not in the US or in the EU, because they got their own licensing exams in the EU for you to pass. So that is the thing. So entrepreneurs or startups, if you're bold enough, if you are not like me, if you are all really young, and if you are really bold enough, you can fill in the gap areas, which I will show you the gaps. If you are smart enough, you can drive in the goal inside. The quality feed is lacking in India. So it is, of course, I can advise you to start build a factory or set up a factory to produce a pelleted feed like you do have or seeing in for cattle and other livestock and for dogs. You can set it up, but the thing is that we don't have any farm, and all these feeds, basically these feeds we are importing actually. For Sri Chitra, we had been importing from France, and now we are finding it difficult because of the COVID, the pandemic, as well as the prices are sky high. And within India, there are lots of feed companies, maybe a handful of them, maybe 3 or 4 feed companies, and they are not really upkeeping their quality. They are compromising on their quality. Why? Because it is a costly affair. For you to maintain the quality of a laboratory rodent or rabbit feed, it is costly. Why? Because you require high crude protein in the case of rats and mice, and a high crude fiber in the case of rabbits. So in rats and mice, for crude protein, ideally the multinational companies, they use either albumin, dry albumin or dried albumin or milk powder, whereas Indian manufacturers, to bring down the cost, they use things like fish meal and things like that, which can while you analyze it, it gives you very good values like 21% of crude protein, but the bioavailability of such crude protein from such an origin will not be that good. So the quality of the feed, and during rainy season we come across with what you call mold or moldy this thing, and the quality is compromised, because as per the regulatory agencies, we are supposed to keep a track on the values of the proxenate principles and pesticide levels and contaminants and things like aflatoxin and ochratoxin in the feed, where the Indian manufacturers feed, if you test it with a third party on a blinded setup, you will not be finding it meeting the standards, and the regulatory bodies will frown upon us. So whereas the quality, the feed which we had been importing from France or from the US or from the EU, they are really good ones. They don't have any mold or thing, and they voluntarily will tell that this for this batch of feed we are withdrawing because it doesn't meet the quality standards. The supplier themselves will withdraw a batch and send us a new one. So their quality is really good. But the thing is that the cost is exponentially high. So you can plan to place a quality feed if you can strike a balance between the cost of an imported feed and at the same time ensure the disadvantages of eliminate the disadvantages of the Indian feed. But in Kerala, you should be really cautious to set up any industry in Kerala, as you know about our circumstances and background in setting up of industries and the cost workouts, because we are not in an agricultural state. So the agricultural products, like we use basically use corn as the background material or the base of this feed, which has to be transported from other states to Kerala, and for you to transport, and all the animal facilities, most of them are in North India and or in central India, or at least out of Kerala. So again, you have to transport it back. You have to bring in the raw material, you have to face all the industrial setup issues here, and you have to send it back, and you have to really think before you put your hands into the quality feed, which even if I am advising you to do that. But you can do a market survey and things like that on your own if you are bold enough. So quality animals, we again lack quality animals. Multinational players like Charles River and Jackson's and Envigo, people like that have started their collaborations within India, and they are giving us animals, but the local, they are costly. As I told you, 24 rats from within India, from within Bangalore, last year costed me something like 1,24,000 rupees for 24 rats. For 24 female rats, it costed like that. So it is a good business for you to set up and to start if you do a market survey, and if you can find customers. You will find a lot within Kerala and outside Kerala. If you are a quality player, then you will have a lot of customers, because the cost is too high now with the multinational companies. Now the scenario in both the cases is the cost is too high and cost is too low. And I will tell you another area where you can invest your money if you are bold enough. That is the services on health monitoring. Here, unlike from the past two cases which I have told you, there are no players now. If you can start a sample receiving report offering lab with PCR and serological capabilities, you will be flourishing in India, because there are no labs, absolutely no labs, and there are a few labs which does the thing based upon paper strips and imported ELISA kits. They give results for a few handful of diseases. But if you can set up a PCR based laboratory which can actively screen and offer timely reports, authentic reports, there are no one in India right now who are doing this. So I wish you all the best. Before you exit the presentation, when you start a job, or if you plan to lay your hands on your animals, even for training, you should know about the 3 R's proposed by the eminent scientists Russell and Birch in biomedical research in 1959. So they have propounded the replace, reduce, and refine. Replacement, reduction, and refinement of laboratory animals to be used. So anyone can use laboratory animals. Anyone can plan a surgery or an experiment. Even you can, without any training, we can set up an experiment. But the thing is that you are advised to. But it is the thing is that it is very much difficult to avoid an animal experiment, especially in the position which I am working in, and especially in a position I am seeing you to be working in a couple of years afterwards. Why? Because it is easy to do, but not easy not to do. So you have to thoroughly follow these 3 principles, like replacement. Whenever possible, you need to replace your animal experiment with a literature. You should start your thing with a literature search. That's the ideal thing to do. If the research is already being carried out by someone else, then why again waste animals just to because anyway nowadays you won't be able to if you are truly representing the results, the peer reviewers will immediately reject, telling that you have repeated an experiment unnecessarily. And if you do an adequate literature search, if you have already data available, then why waste resources and precious time? So these things should be considered before you resort to an animal, to replace an experiment. And if you are absolutely convinced that you require such an experiment to be carried out, then the next step you should follow is not an animal experiment, but you should do a cell line or an in vitro study if possible to gather information. For example, it is not possible 100% of the time where you absolutely require animal experimentation, but there are several times where we miss this point and end up in using more number of animals, which is not the right thing to do. Because for example, an eye application is, or an eye antibiotic is new candidate is being proposed by a scientist, and they are asking you to start animal experiment to treat an animal and see whether it is functional or not. It will be maybe 10 animals, 12 animals. You are starting your experiments right away without doing the literature search one, not doing an in vitro study. Because here, if you had done an in vitro study, I'm just telling a hypothetical scenario. If you if you did an in vitro study, and if you dropped that antibiotic over a cell layer of corneal layer of cells, you are finding out that lots of cells are dying, and then you are again asking the scientist to go back and check what happened, and he or she again went back to her or his lab to see that the pH was extremely acidic or extremely basic. So she corrects it back, and thank you, doctor. And again, you're dropping it on this next layer of cells. You're finding that nothing happens to the cells, and you are carrying out the animal experimentation, which means you have replaced number of animal sufferings, lots of suffering in these animals. You have also reduced the number of animals used. So it has a wide implication, replacement. And the order is important to follow. The 3 R's doesn't mean that you can announce the replacement last and reduction second. So first is to replace, and then after all these exercise, you are convinced that you are supposed to do an experiment in animals, then you are advised to do it with a maximum lower number of animals as possible. At the same time, it shall fetch statistically significant results. So you should be well versed in conducting a statistical program known as power analysis or sample size analysis based upon the previous data or somebody else published data or your pilot project data, and applying them on an equation and finding the optimum number of animals to be used for you to find a statistically significant result. If you don't do that, you end up using more number of animals, and at the same time, if you go on to publish your results, the peer reviewer good peer reviewers will ask the question, why didn't you do a pre-test sample analysis? And you will be held responsible. So it can reduce the number of animals to quite a lot. And refinement comes with training, education, experience, everything, reading, online training, whatever it could be. So you are supposed to refine the experiments. Why? Because a refined person, as a trained person, if you perform maybe 10 oral gavages, 10 of your animals will survive. Whereas if you are not refining your technique with the equipment, which is not a stiff needle which you are using for the oral gavage, you are replacing it with a rubberized one, and then you are refining your technique. You are getting experienced and trained, which means again you are refining your techniques. All the 10 animals will survive. Ultimately, it will have an impact on good data which you are producing, as well as you will reduce the total number of animals used. So it is important for you to learn that animals can be used, of course, but there are these certain principles which are time tested and proven, which we also are bound to follow. So this is our facility. You can visit our facility for a training or something, or as a casual visit, maybe not now after the COVID and after the revamping. So this is one of our environmentally controlled rooms. And so this is all from my side. Maybe I took how many minutes? Almost an hour I took, I think, or even more. I think 2 hours I took. I don't know. So if you got any questions, you can ask me, or you can write to me at harikrishnan sctmsd.ac.in.
Thank you, sir. So we have some questions in the chat box.
Yeah,
Can
please.
you please?
Yeah. Okay. Where shall I see?
Yes, if it is possible, please.
Chat box. Where is the chat box?
So it's
Really
in the
mute.
more, sir, more section, more. Chat's written.
Yeah, the first one. Okay. First one.
You can scroll up, sir, if it is.
Oh, there are a lot of questions.
Yes, sir.
So I hope the session is audible to all. So that is not a question to me. Thank you, Ajit, for that. Questions received. So the institutes in Kerala mainly involved in biomedical research. Postgraduates in which all veterinary and animal science subjects are preferred in such biomedical research institutes. Lab animal, PG, available in India. So I think I have covered some of these. In the institutes in Kerala mainly involved in biomedical research is basically Sri Chitra Tirunal Institute is one, and Rajiv Gandhi Center for Biotechnology in Trivandrum is another one. Now IISER, Valiamala, it is a bit remote but it is still Trivandrum. IISER Trivandrum, I think IISER Trivandrum is having an open post. You can contact someone in IISER Trivandrum or write to me. Maybe I can give you the person to be contacted. I can give you with their with his email address so that you can write. So they are looking for basic BVSC graduates. Me myself joined as a BVSC graduate, and many of the projects here announced as temporary veterinary surgeon and all are veterinary graduates. And I can say that as a one-word answer to which all post graduations are preferred, I can tell in one word that pathology is an important area where across India there are vacancies, and also surgery and anesthesia also are important. And more importantly, nowadays biotechnology is picking up, and because if you have surgical capabilities, and if you are working to produce a transgenic mice or something like that, you need how to play with genes and knockouts and knock-ins and things like that. Then you have an opportunity in the coming or in molecular genetics. Microbiologists have good chances. So these are basically the areas which you should be looking forward for. But don't get disappointed or disheartened. Even if you don't belong to any of these specialities, you can also try your luck with your basic BVSC degree. There are lots of opportunities because these institutes they prefer to open up a position with basic graduates, BVSC graduates, and some institutes, if it is an academic positions, they look even for some PhDs. Scope of animal welfare and behavior. Sorry. Animal welfare as such, for you to study, there is not at least in India there is no such a scope. But for you to do your higher education in animal welfare and behavior, there are lots of scopes. So as I told, there are lots of research groups working for the betterment of environmental conditions, for the betterment of housing standards, to find the truth behind animal the housing standards, their provisions which we are giving to them. There are lots of groups working. If you can find some of these groups and apply for a postdoc or a PhD there, or even for an MSc, it can be of scope. But if you join an Indian institute and if you can find funds, it is a very good area to work with in animal welfare and behavior. Yes, it is possible. So is establishment of private lab animal breeding stations possible in India? It is highly possible. That's what I was. That was what I was telling in my almost my one of those last slides in which I was telling that it is highly profitable, but you should be able to find the market. I can tell that some people are finding it hard to find good mice and rats within Kerala itself, and outside of Kerala it is highly possible. It is a prospect, but especially if you can put a screening lab, or you can attach your screening capabilities to some already set up labs of veterinary origin or of human origin, and if you can offer results, then that could be really wonderful. There will be lots of customers for you from across India, because there is no lab in India as far as I am concerned. We also have a microbiology lab, but unfortunately the thing is that timely reports are a must. If there are 20 or 30 animals reaching your facility for testing, and during the quarantine itself, you should be able to produce a health report of all of them. It shall contain a panel of agents, and each agent, maybe 20, 30 pathogens are to be screened as per FELASA guidelines. And if you go to the FELASA website, you can read more about the health monitoring working group reports, and you can download it free of cost. So they will tell you what are all the parasitic diseases, what are all, most importantly, the viral, the Mycoplasma pulmonis, and lots of other diseases, Hantan, Kilham, Sendai virus, bacteria, lot of bacteria, like starting from Salmonella to a wide range of Citrobacter rodentium, and lots and lots of bacteria and virus has to be screened. Ideally, if you have to ensure your personal safety, and also the authenticity of your data. So this is lacking in India. This is not yet operational in India or fully functional in India, and you can try your luck there. For sure, you will be able to find the most success in this area. Could Dr. Harikrishnan elaborate on how he got into SCTMSD just a few months after BVSC? For instance, on the criteria, whether an exam is involved or whether it is from an interview, whether the candidates. Okay, so that is an interesting question and a nice history for me to rewind and chew the cud. So after maybe a few months I was working, and my area of interest was surgery, and I had been participating in many surgery and field wise laparotomies and these kind of tumor removals and cesarean sections and all. And that was when I came across with a job opportunity or a temporary position in Sri Chitra Tirunal for a coronary stent evaluation program, which I mentioned in depth and detail in one of my first slides. So having seen that, I didn't have any experience. I didn't have any previous experience in the field. I didn't have any previous certificates or courses done in the field. I didn't have any master's education in the field. I didn't have enviable OGPA as well. My OGPA is something like 7.1 or something like, yeah, exactly 7.1 is my OGPA. So, but I tried my luck. I just walked in, and it was a walk-in interview. There was no written test. And that was a time in 2003 where this field was really starting to enter to the log phase, where there didn't have, at that time, luckily for me, they didn't offer or have any written test, but the interview was there, and I couldn't even answer the question, what is the most famous thing from Sri Chitra Tirunal Institute was being asked to me. I didn't even answer that, but somehow I answered many other questions and could get into that. Actually, Sri Chitra Tirunal Institute is famous for its heart valve, its prosthetic heart valve. It's the one and only kind of the prosthetic heart valve, indigenous prosthetic heart valve, and it has crossed over 1 lakh implantations in human patients until now. And the first patient is yet still surviving, and we are exporting it to many countries. I didn't know about that at that time, and I am sure that many of you also, for many of you also, it will be a new piece of information. So I was in your very same shoes in 2003, which was 16 or 17 years ago, but I somehow found this job. It is not. Is MBSc necessary to be a scientist in the field of biomedical research? Actually, I joined as a scientist B with a BVSc. We are all doctors. So at that time, it was not a mandatory. It was not mandatory at all. And in private organizations, if it is contract research organizations, they are absorbing pathologists like anything. If you see Hyderabad, there are lots of veterinary pathologists just working in the field of these drug developing or contract research organizations as pathologists basically. But it is not a mandatory for you to run an animal facility. If you are just having a BVSc, you can try your luck. And it is highly variable that how these institutions announces for new vacancies, whereas I'm not sure whether Sri Chitra Tirunal will again announce it with an MBSc or with the same old BVSc with which I got into. So I think you will be answered by Anandu because he will be posting the recorded video in the Facebook page of yours. Okay, so why does some rats show cannibalism? Is there stem cell? So stem cell research, there is a group working in CTIMST, and I don't think that we are working much into stem cell research in Sri Chitra Tirunal, but in Rajiv Gandhi Institute they are working a bit on or more on stem cells. I think so. Here are groups, and I have shown you the website. There is a PhD available. If you are an MBSc scholar, you can look also for PhD with some funding is also available in Sri Chitra Tirunal Institute, and it is getting announced twice per year. You can keep an eye on the website. So why does some rats show cannibalism? Rats actually basically mice fight with each other. I should tell you in depth about what happens in mice. In the wild, they show highly territorial behavior. Like the dogs, they mark their territories. The rats also does that, but more about mice. The male mice show territorial behavior, and they live as societies. And in a society or in a group, there won't be more than one male mice. But in laboratory animal science or in the practice, what we are doing is that we don't want unwanted births. We don't want our research animals to be pregnant. We want a planned breeding, so that we sex the cages. We keep all females in a cage and all males in a cage, which is totally against the what we are seeing in the wild. So that could be the reason why they fight with each other. And in some strains which are wild strains like C57BL bar 6J is one dark strain with dark hair coat, and they fight with each other and they kill each other. And some breeding animals, they eat their young ones, and it could be a part of their natural poor maternal ability. It could also be a part of their nutritional inadequacy, and it can also be a part of their habits. And usually after the first time parturition, or from the second time onwards, they lose this habit of cannibalism. It is really easy for you to get into the applied field of biomedical research, because the job vacancies are more in the applied field of biomedical research. You can enter as a BVSc scientist who are doing surgeries, because you can prescribe drugs. You are easily trainable. Many of the contract research organizations and many of the drug manufacturing companies, they will absorb you, and most of these these agencies, these industries, those are in the already described states like Gujarat, Ahmedabad, Hyderabad, Bangalore, Chennai, etc. So nutritionist, yes, of course. As I told, feed manufacturing companies are there a lot, not a lot, but a handful. But I don't think that there are lots of scope in for nutritionists. But you can try your luck in any of these general categories and find whether you are in particular lucky to find a nutritionist job vacancy. Can a BVSC graduate who underwent the training you mentioned, or a PG graduate in surgery without such a training, has better opportunity? Better opportunity is for everyone. If you are a BVSC graduate, the world is wider for you. So after finding each degree, getting each degree, you are getting yourself narrowed down. I'm not discouraging the MVSC graduates. I'm also having a master's, but the thing is that if you apply for a job, like you cannot go back and apply for a broader job. Like if I lose my job here, then I will have to start from scratch to find myself as a practicing vet by assisting some other vets, get the old lessons learned again, brush up my old pharmacology course and everything like that, and get the doses of the drugs correct. You have to do a kind of go in the reverse direction to find yourself suit again. But as far as a BVSC graduate is concerned, the world is wider. You can try your luck with laboratory animals. You can try your luck with animal breeding control program. You can do many things. But it is always good if you plan to work in or try to find opportunities in the field of laboratory animal science to at least go through those free websites which I gave you, read a bit, and go to the CPCSA website and find the institutions registered and find your own associations and ways to speak up for you to create more posts on a whole as in collective and in person to keep an eye on everything and find your job. What is a pharmacologist? So pharmacologist has lots of job, but in laboratory animal management, you are as good as a BVSC graduate. So it is an applied field of biomedical research that your specialty will count for you. But as far as laboratory animal management is concerned, there is no added advantage that you are a pharmacologist. So I hope I have answered most of the questions which you have, but you are free to type to me in this address. I can whenever I can find my time, I'll punch back my answers.
Sir, shall we wind up the session?
Yes, go ahead, please.
Okay. 7:45. Okay, thank you, sir. Thank you for this wonderful session. And I thank everyone on behalf of Kalpagam IVRA for the session, and also for IVA Kerala for providing this platform. Thank you, sir. We'll see you again.
Thank you so much. Thank you so much. Bye.
Okay, thank you, sir.
Hello, sir.
Hello, Anandu.
Sir, wonderful section. Elaborative ആയിട്ടുള്ള class ആയിരുന്നു. Yes, sir. എല്ലാവർക്കും നല്ല ഉപകാരപ്പെടുന്ന ഒരു ക്ലാസ് ആയിരുന്നു. ഞാൻ ദയമോൻ സാറിനോട് താങ്ക്സ് പറയുകയാണ് കൽപ്പകത്തിന്റെ പേരിൽ. സാർ ജോയിൻ ചെയ്തിട്ടില്ല, ഞാൻ സാറാണ് സാറിന്റെ നെയിം സജസ്റ്റ് ചെയ്തത്.
താങ്ക് യു സോ മച്ച്. എനിക്ക് ഫീഡ്ബാക്ക് അറിയേണ്ടത് ഇവർക്ക് അത് പ്രയോജനപ്പെട്ടോ എന്നുള്ള ഒരു
Yes, sir.
ഫീഡ്ബാക്ക് ഒരു മെയിൽ വഴി എന്നെ അറിയിച്ചു കഴിഞ്ഞാൽ എനിക്ക് യൂസ്ഫുൾ
Sure,
ആയിരിക്കും.
sir. Sure, sir. ഞാൻ ഫീഡ്ബാക്സ് ഞാൻ കളക്ട് ചെയ്യുന്നുണ്ട് എല്ലാ ക്ലാസ്സിന്റെ അവസാനം, അത് ഞാൻ അയച്ചു തരുന്നത്.
ഓക്കെ ഓക്കെ, താങ്ക് യു.
PDF ഞാൻ എല്ലാവർക്കും മെയിൽ ചെയ്യാം.
Sir,
ഞാൻ
already
അയച്ചു തരാം.
ചാൻസില് കുറച്ച് വന്നിട്ടുണ്ട്.
എന്തോ
കുറച്ച് ചാറ്റ്സിൽ കുറച്ച് അഭിപ്രായങ്ങൾ 2-3
അത്
ചാറ്റിൽ
വന്നിട്ടുണ്ട്.
കുറച്ച് ഫീഡ്ബാക്സ്
ഫോർ ദം.
ശരി ശരി.
ചാറ്റ്സിൽ കുറച്ച് ഫീഡ്ബാക്സ് വന്നിട്ടുണ്ട്.
താങ്ക് യു. അപ്പൊ ഇതിന്റെ റെക്കോർഡ് സെഷൻ എവിടെ കാണാൻ പറ്റും ആനന്ദു?
സർ, ചെസ് ഫോർ വെച്ച് എന്ന് പറഞ്ഞ ഫേസ്ബുക്ക് പേജ് ഉണ്ട്, അതിൽ അപ്ലോഡ് ചെയ്യുന്നതായിരിക്കും.
സാറിന് അതിന്റെ ലിങ്ക് അയച്ചു തരാം.
ലിങ്ക് അയച്ചു തരാം സർ.
ശരി, താങ്ക് യു സോ മച്ച്. നിങ്ങൾ ഒരുപാട് എഫേർട്ട് എടുക്കുന്നുണ്ട് ഇതിനു വേണ്ടി. നല്ല കാര്യം.
താങ്ക് യു സർ.
Thank you, sir. Shall we leave, sir?
Why not? We will keep in touch.
Thank you.
Thank you so
Yes,
much.
sir.
Thank
Thank you very much.
Thank you, sir.
Thank you.